It's the most common death in the United States, more common than cancer. 1 in 5 of us all will ultimately die of sudden cardiac death. Okay, so sudden cardiac arrest is relatively common compared to other types of diseases. It's not just athletes. You all know this very, very well. We tend to think of this as happening to athletes who collapse on the field or collapse on the basketball court. It's not just athletes. 2 out of every 3 sudden cardiac arrests in children and young adults will be in non-athletes or will occur outside of sports. It's a big number. This is the number that kind of keeps me up at night. 2 out of every 3 children who die of sudden cardiac arrest have no symptoms. And it can occur at any age. So for example, in our research lab, we study all the way down to children who die less than 1 year of age, which we call sudden infant death syndrome. Now to be sure, sudden infant death syndrome is not all the heart, but we're interested in the full spectrum of disease. So just because a child dies who's 2, 3, 4, 5, or 6, doesn't mean it can't be a sudden cardiac arrest.
Has there been a link found between SIDS and sudden cardiac arrest?
Like
family history of SIDS, does that have a higher?
There is a link. The link doesn't explain every baby who dies, but there is a link for some families, yes. So we'll start with the very, very basics. What is sudden cardiac arrest? So the heart is the pumping chamber for blood in the body. There's 2 sides of that heart, the right side and the left side. The right side pumps blood to the lungs, the left side pumps blood to the body. And for the heart to do this, it has to listen to lots of different competing demands. Are you standing up or are you sitting down? Are you sleeping or are you running? Are you eating or are you exercising? So the heart is very attuned to what your body needs and delivers just the right amount of blood typically for what your body's demands are. Sudden cardiac arrest is any time that blood doesn't get out to the body. And the heart can literally stop, or more frequently, the heart goes into a misbeat, which causes the heart to not pump so ineffectively that the blood doesn't get to the body effectively at all. So my first recommendation for you all to consider is, as you go through this process, I think any family who's experienced sudden cardiac arrest in their family should seek an expert in sudden cardiac arrest. I think there's a lot of individuals who might have a wandering path to finding an expert, and that's fine. But you should always try to find an expert. The reason being, and approximately 2/3 of them will have an explanation we find at autopsy. And the majority of the time, if there is something that's found at autopsy, usually it's a muscle disease of the heart or a coronary artery disease of the heart. And then there are a third, where even after a regular autopsy, there is no identifiable cause. Those are the ones where we start to think about arrhythmias, about channel disease, about electrical disease, because remember, those defects are ion channels, they're molecular, they're very small. And so a typical autopsy, where they look at the heart, they even look at the cells of the heart, is not enough to see those defects. So these are the pumping chambers. Pumping chambers are big. The walls that separate them are normal in size. And you can see over here where the arrow is pointing, the muscle disease of the heart there. And this is called hypertrophic cardiomyopathy, where the walls are thicker. So you can imagine how hearts, like every muscle, get thicker if they need to. Right? So if you go and you work out at the gym every day, your muscles are going to be bigger. Your heart can do that if it needs. But imagine if your heart, with no trigger at all, got too thick. And nothing stopped it from getting thicker and thicker and thicker. That's hypertrophic cardiomyopathy. When athletes collapse and die, it's the most common cause of sudden cardiac arrest in athletes. It's thinking about the heart, just like this. So to the heart, and they give cardiologists a view of the walls, windows, structure of the heart. And you can see sometimes the heart's very, very thick. This is hypertrophic cardiomyopathy, where the heart's too thick. So all of us have about 3 billion A's, G's, C's, and T's that make up our DNA, that make up our gene. And this defines who we are. It also defines diseases like hypertrophic cardiomyopathy. And when our cells are dividing there's always changes that are made. That's what makes siblings different, that are not identical twins. Sometimes these changes can occur just in the wrong spot. And if it occurs in certain spots in the DNA, it can cause these diseases. And all it takes is one misspelling out of 3 billion nucleotides to find it. So you can imagine how this can be a challenge to find, to diagnose. If you're looking in the genome, trying to figure out what happened to families like yours, it only takes one misspelling of 3 billion nucleotides. And we're getting a lot smarter about how to do this, but that's the challenge. The problem is not in the bricks and mortar. The problem is in like the sand that made up the concrete, that made up the mortar, that made up the walls of the house. Very hard to find. And so if you take a healthy human heart, you take a small little piece and you look at the individual cells, which is what the medical examiner does, you still can't see it. You still won't be able to see it. You have to go down to the molecular level. These little things, tiny little molecules, whose whole job it is to move salts from the outside of the blood, outside of the cell to the inside of the cell, or vice versa. Single file salt molecules. That's where arrhythmias, channelopathies lie. Not in the muscle, but in these ion channels. Does that make sense? Manifests on the ECG where the QT interval, we'll talk a little bit about that, takes a little bit more time to happen in the heart. So it looks long on our test, long QT. The trouble is that machines, when they read an ECG, they don't measure that number very, very well. The way the heart makes that QT, it's different for everybody and changes over time. And so computers are very bad at figuring out what the number is. And oftentimes the machine says, oh, it's too high, when in fact it's not. And so I see a lot of families, just like yours, where the machine read it too high, and then when you measure it correctly, it's not long at all.
Okay.
But in a family with fainting, other questions come up. When did the fainting occur? Probably deserves another closer look. And if the QT is normal, then we should probably look for other things as well. Doesn't mean we're going to find something, doesn't mean you have a diagnosis, but it means we should have a conversation. You should try to be heard about what's going on with your history. There are some tests on occasion that we can do, but typically the only blood work that we do in my clinic is genetic testing. Certainly if the salts in your blood are messed up, like you're dehydrated, so maybe your potassium, like for eating bananas, is too low in your blood, or your magnesium is too low in your blood, that can precipitate some of these events. So some of our patients with long QT, like, you better eat a banana every morning. And if you should stay hydrated, and if you get sick and you can't drink, I need you to let us know. Because those salts in the blood can make them sicker, but there's nothing that tells you what the diagnosis is in the blood, outside of the DNA. Great question. Tracks, and that little squiggle after is where your heart resets electrically, the bottom chambers. And you wouldn't think that that's a big deal if your heart takes a little bit longer to reset, but it can be. And people with long QT syndrome, their heart does this every time it beats. And all it takes is one misbeat at the right time over a person's lifespan to cause them. Over here, this thing kind of looks like ribbon that's been twisted up, is actually a three-dimensional molecular structure at the atomic level of a potassium channel. It's got a very kind of boring name, KV11.1, but one misspelling in this potassium channel is the cause of long QT type 2. All it takes is one misspelling in the right place of this ion channel. Now, to make things a little bit complicated, why don't we do just genetic testing on everybody? Many, many people, in fact, people in this room who don't have long QT probably have misspellings in this ion channel too. Right? That's what makes us who we are. We're all different in some ways. But it takes only one in the right spot of the ion channel, of the right kinds, in the right context to cause disease. And what happens is, because the heart takes a little bit longer to reset, if an extra beat comes in, and we have these extra beats all the time, but if that extra beat happens at just the right time, in just the right kind of way, it can cause that squiggly line. When we saw that earlier, that's the most solid point. A arrhythmia that, if it doesn't stop on its own, can be life-threatening. So that's how a single misspelling in an ion channel can cause a shift in milliseconds of how a heart resets and how that predisposes to it. Questions about this? This is not easy stuff. This is pretty high level stuff. For example, if a healthy child faints and they get an ECG right after that child's fainted, like they're in the ER and they're being watched, the QT can be long. And it doesn't mean they have long QT syndrome. But I would really want to make sure that we talk about exactly what happened, and there's other testing we can do. So it's possible, and I can't tell you without knowing a lot more information and knowing what testing, but it is possible. It is. So these are the kinds of diseases you may hear. Again, these are all electrical, so all the testing is going to be these squiggly line tests. There's long QT, or the T waves out there, it takes longer to reset. There's also something called short QT, and this is very rare. You see how close that T wave is? How close it is to this squiggly line? Here you've got, it's really far away. So you don't want to have a T wave that's too far away or too close. You want to have it just right. So like Goldilocks and Three Bears, right? Regatta, yeah. This is what we call a regatta pattern, and it can be very hard to see. It can sometimes be seen only in certain circumstances and only in certain areas of the heart. Where it goes up, goes down, comes back up again, is regatta. CPVT, which is catecholaminergic polymorphic ventricular tachycardia. It's got a very long name, but this is the most deadly arrhythmia that we know about. So if you have CPVT, the chance of having a sudden cardiac arrest before the age of 20 is 50%, which is staggering. And then if we don't know what it is, but we know there's a nasty arrhythmia, sometimes we call it idiopathic ventricular fibrillation. Idiopathic just means unknown, but we know we've got an arrhythmia there. Why it's there, we don't know.
QT. With long QT, it can be,
Yes,
it's usually hereditary, but there can be times that it's not.
Great question. Yes. Yes. So there's lots of reasons for that. Sometimes with some of these misspellings, so channelopathies are misspellings too. Sometimes those misspellings occur in a way where it just happened to that one child. And both parents don't have it, and the rest of the family doesn't have it. Exactly. And so just like heart and muscle diseases, cardiomyopathies, channelopathies are genetic, with those same misspellings, just in different parts of the heart. Yes,
The
ma'am?
channelopathies, are those found by genetic testing? Okay.
Yeah, exactly. So you can imagine how if we can't see it by imaging, and sometimes we can see it by our electrical testing, genetic testing is the best test that we have. And this is a subtle thing, but it's an important point. And we'll talk about it in a little bit. The genetic testing should never be done to make the diagnosis. You should see somebody who says, I think you have this disease, or I have a high suspicion, so we're going to get the genetic testing. Because the genetic testing is most impactful for the family. So if you can find the mutation, if you find that spelling error out of the 3 billion letters in the genome, you can look for other people in the family who don't have any sign of it when they have their testing. The genetic test goes, I don't know. And this raises a few points. Just as a cardiologist should never trust the machine read of how long that QT is, we talked about that. A cardiologist should never trust the genetic test report that comes out. And if it's misinterpreted, these things change over time. We talked about where our knowledge of this field is growing all the time. So there are definitely variants, mutations that change, and we don't know enough about them to know if they're pathogenic or not. This one's uncertain. And so, if my son had an arrest, we had a variant of uncertain significance, I'd want to know, how certain are you, as the cardiologist, that this variant is pathologic? We talked about, if it is, we can screen the family. But if we got it wrong, if we made the wrong decision, and it turns out it has nothing to do with the family's disease, then we might start doing things like treatments that we don't need, anxiety that we don't need in the family, ICDs that don't need to be implanted in the family. So the first question I would ask is, how certain are you that that variant, whatever it is pathologic or not? How certain are you? And that provider should be able to say, because I think of it as this, this, and this, and this, not because that's what the paper said. If that's what the paper said, that might be a warning sign. We do a genetic reevaluation. So if you come to us with a genetic test, or if that genetic test is done in clinic, we reevaluate the pathogenicity of that variant. I don't care what the report says, pathogenic, not pathogenic, unknown significance, I do it myself. There are certain criteria we can use that are research-based to support or deny those things. Does that make sense? Just like you don't test that ECG right after you faint, don't test the genetic test, or don't trust the genetic test on its face. And the provider should not either. It's meaningful as well.
Do you recommend ICDs for everyone with long QT?
No.
Okay.
Certain patients under certain circumstances. And certainly patients with just a genetic variant, just a mutation but have no evidence of disease, no. Good question. But your gene test is negative, you still have long QT. You just don't have a gene that's been identified yet. We don't know enough about it. Okay. Having a negative genetic test in a patient with disease never means they don't have disease. Diagnosis first, gene test second.
Didn't know that.
Okay? Now, this is important. We don't know all the genes, all these spots in the genome that can cause disease. So if you have 10 patients with hypertrophic cardiomyopathy, we may only get a positive genetic test back in 4 of them. Those other 6, they still have disease. We just don't know enough about the disease to know why. And thus, we can't find something to look for in the family. So maybe genotype negative. 3 out of 10 long QT, 7 out of 10 Brugada will be genotype negative. And a negative genetic test never excludes the possibility of disease. Right? You made your diagnosis. How can it? So, one of the things that on our research side, so I did a lot of extra training, avoiding getting a real job for as long as I possibly could. But one of the things that I'm very passionate about is not just the patient care, but trying to advance the field. You saw all those numbers about people who we don't know why they have disease, all those genotype negative individuals. So one of the things that we're very proud of is the fact that we take families whose gene test is negative, but they've got disease, and we say we can try to figure this out. And we use a number of additional tools and the research lab. And every now and then, it doesn't happen for all the families, every now and then though, we're able to find a genetic cause that's never been described. If we can understand it in the family, we might be able to use it to help other individuals with that disease. And so this is an article of a collaboration that we're a part of that came up in Wired. You can take a look at it. It's got some somewhat overly dramatic pictures of it, but like heart disease, cardiomyopathies, heart muscle disease, and channelopathies. We're talking a lot about that today. I recommend you be evaluated by a sudden cardiac arrest expert. I recommend you think about genetic testing with your provider. Or remember, make your diagnosis first, gene test second. Because the person who gets the first genetic test should be affected. A positive test serves as a marker for the family. A negative test never rules out disease. And means you don't have that marker. So you can't say, well, everything is fine. Doesn't mean that you should be followed every 6 months or every year, but they should always be followed, always in the back of your minds. So I'm part of a great big team at Duke. You might remember